Over the last 10 years or so, genome-wide association studies and sequencing have identified up to around 60, 70 loci associated with Alzheimer's disease. Those studies have revealed a surprise. Initially, it was a surprise that these genes are expressed by the innate immune system. And at the beginning, it was a bit difficult to reconcile how innate immune genes would function along the amyloid cascades. But a lot of work has gone on over the last 10 years to understand the function of TREM2, CD33, those types of genes, to understand how microglia are actually tied into the amyloid cascades. Yeah, so with the GWAS studies that have identified risk genes associated with Alzheimer's, a lot of them expressed by the innate immune system, what we wanted to do was understand how these risk genes are connected and how they fit into the amyloid cascade. So are they involved in responding to the amyloid beta plaques? Are they responding to the tau? Are they responding to both or other stages of the amyloid cascade as well? And so the study that we performed to address that question was to use transgenic mice that either have amyloid pathology or tau pathology, and then to do RNA sequencing to look at gene expression associated with those pathologies. And then we used a bioinformatics tool called network analysis. Basically, what that does is it mines the RNA-seq data to identify co-expression networks, so basically genes that behave the same way. And so we had a collection of genes that respond to amyloid and a collection of genes that respond to tau. These networks that responded to actually both amyloid and tau were expressed mainly by the innate immune cell, so by the microglia. And then the second element of the study was to compare that with genome-wide variation, so looking at GWAS data associated with Alzheimer's. We saw a significant enrichment of genes that are associated with Alzheimer's in the network that was responding to amyloid, but not to tau. And so to us, it tells us that the amyloid-responsive microglial network is responding to amyloid, and it's regulating the step between amyloid pathology and tau pathology. Even though there was a response to tau as well, that network wasn't enriched in risk genes associated with Alzheimer's. So genes associated with Alzheimer's risk are less likely to be regulating that step, although other people have shown since that microglia also do respond to tau pathology as well.